Hello and welcome dear students. Today we are going to discuss about the topic sequencing strategies, that is PCR based Sanger sequencing to exosome sequencing. The main objectives of today's lecture are to know the basics of sequencing strategies, to discuss about Sanger sequencing, to discuss about exosome sequencing, to understand the comparisons of exosome sequencing to other approaches, to discuss about whole genome sequencing, to know the applications of sequencing strategies, and finally to understand the clinical diagnostics of sequencing strategies. Dear students, let's start with the introduction first. Sequencing is the process of determining the sequence of nucleotide bases A's, T's, C's and G's, that's adenosine, thymine, cytosine and guanine in a piece of DNA. Today, with the right equipment and materials, sequencing a short piece of DNA is relatively straightforward. Sequencing an entire genome, all of an organism's DNA, remains a complex task. It requires breaking the DNA of the genome into many smaller pieces, sequencing the pieces, and assembling the sequences into a single long consensus. However, new methods that have been developed over the past 2 decades, genome sequencing is now much faster and less expensive than it was during the Human Genome Project when it started in 1990s and was completed in 2003. Sanger sequencing is a method of DNA sequencing that involves electrophoresis and is based on the random incorporation of chain terminating dideoxynucleotides by DNA polymerase during in vitro DNA replication. More recently, higher volume Sanger sequencing has been replaced by next generation sequencing methods, especially for large scale automated genomes analysis. However, the Sanger method remains in wide use for smaller scale projects and for validation of deep sequencing results. It still has the advantage over short read sequencing technologies like Illumina in that it can produce DNA sequence reads of more than 500 nucleotides and maintains a very low error rate with accuracies around 99.9%. Sanger sequencing is still actively being used in efforts for public health initiatives such as sequencing the spike protein from SARS-CoV-2 as well as for the surveillance of norovirus outbreaks. Exosome sequencing is a process that's used to determine the nucleotide sequence primarily of the exogenic or protein coding regions of an individual's genome and related sequences representing approximately 1% of the complete DNA sequence. Exosome sequencing focuses on target sequencing of the protein coding regions of the genomic DNA and shows promise as a new tool in gene discovery for complex diseases and for facilitating the accurate diagnostics of individuals with unresolved or unsolved Mendelian conditions. Dear students, let's now understand the Sanger sequencing in detail. Regions of DNA up to about 900 base pairs in length are routinely sequenced using a method called Sanger sequencing. Sanger sequencing was developed by the British biochemist Fred Sanger and his colleagues in 1977. In the Human Genome Project, Sanger sequencing was used to determine the sequences of many relatively small fragments of human DNA. The fragments were aligned based on overlapping portions to assemble the sequencing of larger regions of DNA and eventually the entire chromosome. although genomes are now typically sequenced using other methods that are faster and less expensive, Sanger sequencing is still in wide use for the sequencing of individual pieces of DNA, such as fragments used in DNA cloning or generated through polymerase chain reaction. The requirements for Sanger sequencing are: Sanger sequencing involves making many copies of a target DNA region. Its ingredients are similar to those needed for DNA replication in an organism or for polymerase chain reaction, which copies DNA in vitro. They include a DNA polymerase enzyme, a primer, which is a short piece of single-stranded DNA that binds to the template DNA and acts as a starter for the polymerase. The 4 DNA nucleotides, that is the DATP, DTTP, DCTP, and DGTP. The template DNA to be sequenced. However, a Sanger sequencing reaction also contains a unique ingredient, that is dideoxy, or versions of all 4 nucleotides, that is DDATP, DDTTP, DDCTP, and DDGTP, each labeled with a different color of dye. Dear students, let's now understand the method of Sanger sequencing. The DNA sample to be sequenced in Sanger sequencing is combined in a tube with primer, DNA polymerase, and DNA nucleotides. The 4 dye-labeled chain terminating dideoxy nucleotides are added as well, but in much smaller amounts than the ordinary nucleotides. The mixture is first heated to denature the template DNA, that is separation of the strands, then cooled so that the primer can bind to the single-stranded template. Once the primer is bound, the temperature is raised again, allowing DNA polymerase to synthesize the new DNA starting from the primer. DNA polymerase will continue adding nucleotides to the chain until it happens to add a dideoxy nucleotide instead of a normal one. At that point, no further nucleotides can be added, so the strand will end with the dideoxy nucleotide. This process is repeated in a number of cycles. By the time the cycling is complete, it's virtually guaranteed that a dideoxynucleotide will have been incorporated at every single position of the target DNA in at least one reaction. That is, the tube will contain fragments of DNA lengths ending at each of the nucleotide positions in the original DNA. The ends of the fragments will be labeled with dyes that indicate their final nucleotide. After the reaction is done, the fragments are run through a long, thin tube containing a gel matrix. Short fragments move quickly through the pores of the gel, while long fragments move more slowly. As each fragment crosses the finish line at the end of the tube, it's illuminated by a laser, allowing the attached dye to be detected. The smallest fragments, ending just one nucleotide after the primer, crosses the finish line first, followed by the next smallest fragment ending 2 nucleotides after the primer, and so on and so forth. Thus, from the colors of dyes registered one after another on the detector, the sequence of the original piece of DNA can be built up one nucleotide at a time. The data recovered or recorded by the detector consists of a series of peaks in fluorescence intensity. The DNA sequence is read from the peaks in the chromatogram. Dear students, let's now discuss the uses and limitations of Sanger sequencing. Sanger sequencing gives high quality sequence for relatively long stretches of DNA. It's typically used to sequence individual pieces of DNA such as bacterial plasmids or DNA copied in PCR. However, Sanger sequencing is expensive and inefficient for larger scale projects such as the sequencing of an entire genome or metagenome, that is the collective genome of a microbial community. For tasks such as these, new large-scale sequencing techniques are faster and less expensive. Dear students, let's now understand the exosome sequencing. Exosome sequencing, also known as whole exosome sequencing or WES, is a genomic technique for sequencing all of the protein coding regions of genes in a genome known as the exosome. It consists of two steps. The first step is to select only a subset of DNA that encodes proteins. These regions are known as exons. Humans have about 180,000 exons, constituting about 1% of the human genome or approximately 30 million base pairs. The second step is to sequence the exonic DNA using any high throughput DNA sequencing technology. The goal of this approach is to identify genetic variants that alter or change protein sequences, and to do this at a much lower cost than whole genome sequencing, since these variants can be responsible for both Mendelian and common polygenic diseases such as Alzheimer's disease. Whole exome sequencing has been applied both in academic research and as a clinical diagnostics. Dear students, let's have a comparison of exosome sequencing to other approaches of sequencing. Exosome sequencing is especially effective in the study of rare Mendelian diseases because it is an efficient way to identify the genetic variants in all of an individual's genes. These diseases are most often caused by very rare genetic variants that are only present in a tiny number of individuals. By contrast, techniques such as SNP arrays can only detect shared genetic variants that are common to many individuals in the wider population. Furthermore, because severe disease-causing variants are much more likely to be in the protein coding sequence, focusing on this 1% costs far less than whole genome sequencing, but still detects a high yield of relevant variants. In the past, clinical genetics tests were chosen based on the clinical presentation of the patient, that is focused on one gene or a small number of known to be associated with a particular syndrome, or surveyed only certain types of variation, that is for example comparative genome hybridization, but provided definitive genetic diagnosis in fewer than half of all patients. Exosome sequencing is now increasingly used to complement these other tests, both to find mutations in genes already known to cause diseases as well as to identify novel genes by comparing exosomes from patients with similar features. Dear students, let's now do the comparison of exosome sequencing with other technologies. There are multiple technologies available that identify genetic variants. Each technology has advantages and disadvantages in terms of technical and financial factors. Two such technologies are microarrays and whole genome sequencing. Let's first discuss the first one, that is the microarray based genotyping. Microarrays use hybridization probes to test the prevalence of known DNA sequences, thus they cannot be used to identify unexpected genetic changes. In contrast, the high throughput sequencing technologies used in exosome sequencing directly provides the nucleotide sequences of DNA at the thousands of exonic loci tested. Hence, WES addresses some of the present limitations of hybridization genotyping arrays. Although exosome sequencing is more expensive than hybridization based technologies on a per sample basis, its cost has been decreasing due to the falling cost and increased throughput of whole genome sequencing. Dear students, the second one is whole genome sequencing. Exosome sequencing is only able to identify those variants found in the coding region of genes which affect protein function. It's not able to identify the structural and non-coding variants associated with the disease, which can be found using other methods such as whole genome sequencing. There remains 99% of the human genome that is not covered using exosome sequencing. And exosome sequencing allows sequencing of portions of the genome over at least 20 times as many samples compared to whole genome sequencing. For translation of identified rare variants into the clinic, sample size and the ability to interpret the results to provide a clinical diagnosis indicates that with the current knowledge in genetics, there are reports of exosome sequencing being used for assisting diagnosis. The cost of exosome sequencing is typically lower than whole genome sequencing. Dear students, let's now discuss the ethical implications in sequencing strategies. New technologies in genomics have changed the way researchers approach both basic and translational research. With approaches such as exosome sequencing, it is possible to significantly enhance the data generated from individual genomes, which has put forth a series of questions on how to deal with the vast amount of information. Should the individuals in these studies be allowed to have access to their sequencing information? This data can lead to unexpected findings and complicate clinical utility and patient benefit. This area of genomics still remains a challenge, and researchers are looking into how to address these questions. Dear students, let's now discuss the applications of exosome sequencing. By using exosome sequencing, fixed cost studies can sequence samples to much higher depth than could be achieved with whole genome sequencing. This additional depth makes exosome sequencing well suited to several applications that need reliable variant calls. Dear students, let's now discuss about the rare variant mapping in complex disorders. Current association studies have focused on common variation across genome, as these are the easiest to identify with the current assays. However, disease-causing variants of large effect have been found to live within exosomes in candidate gene studies, and because of negative selection are found in much lower allele frequencies and may remain untyped in current standard genotyping assays. So whole genome sequencing is a potential method to assay novel variant across the genome. However, in complex disorders such as autism, a large number of genes are thought to be associated with disease risk. This heterogeneity of underlying risk means that very large sample sizes are required for gene discovery, and thus whole genome sequencing is not particularly cost effective. This sample size issue is alleviated by the development of novel advanced analytical methods, which effectively map disease genes despite the genetic mutations are rare at variant level. In addition, variants in coding regions have been much more extensively studied, and their functional implications are much easier to derive, making the practical applications of variants within the targeted exosome region more immediately accessible. Exosome sequencing in rare variant gene discovery remains a very active and ongoing area of research. To date, few associated genes have been uncovered thus far, but there is growing evidence that a significant burden of risk is observed across sets of genes. The exosome sequencing has reported rare variants in KRT82 gene in the autoimmune disorder alopecia areata. Dear students, let's now discuss the discovery of Mendelian disorders using these sequencing techniques. In Mendelian disorders of large effect, findings thus far suggest one or a very small number of variants within coding genes underlying the entire condition. Because of the severity of these disorders, the few casual variants are presumed to be extremely rare or novel in the population and would be missed by any standard genotyping assay. Exosome sequencing provides high coverage variant calls across coding regions which are needed to separate true variants from noise. A successful model of Mendelian gene discovery involves the discovery of de novo variants using trio sequencing where parents and proband are genotyped. Dear students, let's now understand some case studies. A study published in September 2009 discussed a proof of concept experiment to determine if it was possible to identify causal genetic variants using exosome sequencing. They sequenced 4 individuals with Freeman-Sheldon syndrome, a rare autosomal dominant disorder known to be caused by a mutation in the gene MYH3. 8 HapMap individuals were also sequenced to remove common variants in order to identify the causal gene for FSS. After exclusion of common variants, the authors were able to identify MYH3, which confirms that exosome sequencing can be used to identify causal variants of rare disorders. This was the first reported study that used exosome sequencing as an approach to identify an unknown causal gene for a rare Mendelian disorder. Dear students, let's now discuss the clinical diagnostic of sequencing strategies. Exosome sequencing can be used to diagnose the genetic cause of disease in a patient. Identification of the underlying disease gene mutation can have major implications for diagnostic and therapeutic approaches that can guide prediction of disease natural history and makes it possible to test at-risk family members. There are many factors that make exosome sequencing superior to single gene analysis, including the ability to identify mutations in genes that were not tested due to an atypical clinical presentation, or the ability to identify clinical cases where mutations from different genes contribute to the different phenotypes in the same patient. Having diagnosed a genetic cause of a disease, this information may guide the selection of appropriate treatment. The first time this strategy was performed successfully in the clinic was in the treatment of an infant with inflammatory bowel disease. Researchers have used exosome sequencing to identify the underlying mutation for a patient with Bartter syndrome and congenital chloride diarrhea. Another one is data analysis from sequencing strategies. The statistical analysis of the large quantity of data generated from sequencing approach is a challenge. Even by only sequencing the exosomes of individuals, a large quantity of data and sequence information is generated, which requires a significant amount of data analysis. Challenges associated with the analysis of this data include challenges in programs used to align and assemble sequence reads. Various sequencing technologies also have different error rates and generate various read lengths, which can pose challenges in comparing results from different sequencing platforms. False positive and false negative findings are associated with genomic resequencing approach and are critical issues. A few strategies have been developed to improve the quality of exosome data, such as comparing the genetic variants identified between sequencing and array-based genotyping. So comparing the coding SNPs to a whole genome sequenced individual with a disorder. Comparing the coding SNPs with Sanger sequencing of HapMap individuals. Rare recessive disorders would not have single nucleotide polymorphisms or SNPs in public databases such as DB-SNP. More common recessive phenotypes may have disease-causing variants reported in DB-SNP. For example, the most common cystic fibrosis variant has an allele frequency of about 3% in most populations. Screening out such variants might erroneously exclude such genes from consideration. Genes for recessive disorders are usually easier to identify than dominant disorders because the genes are less likely to have more than one rare non-synonymous variant. The system that screens common genetic variants relies on DB-SNP, which may not have accurate information about the variation of alleles. Using lists of common variation from a study exosome or genome-wide sequenced individual would be more reliable. A challenge in this approach is that as the number of exosomes sequenced increases, DB-SNPs will also increase in the number of uncommon variants. It will be necessary to develop threshold to define the common variants that are unlikely to be associated with a disease phenotype. Genetic heterogeneity and population ethnicity are also major limitations, as they may increase the number of false positive and false negative findings, which will make the identification of candidate genes more difficult. Of course, it is possible to reduce the stringency of the thresholds in the presence of heterogeneity and ethnicity. However, this will reduce the power to detect variants as well. Using a genotype first approach to identify candidate genes might also offer a solution to overcome these limitations. Unlike common variant analysis, the analysis of rare variants in whole exome sequencing studies evaluates variant sets rather than single variant. So, functional annotations predict the effect or function of rare variants and help prioritize rare functional variants. Incorporating these annotations can effectively boost the power of genetic association of rare variant analysis of whole genome sequencing studies. Some methods and tools have been developed to perform functionally informed rare variant association analysis by incorporating functional annotations to empower analysis in whole exome sequencing studies. Dear students, let's now conclude the lecture. DNA sequencing is the process of determining the nucleotide order of a given DNA fragment. So far, most DNA sequencing has been performed using the Sanger method developed by Frederick Sanger. This technique uses sequence-specific termination of a DNA synthesis reaction using modified nucleotide substrates. The sequence of DNA encodes the necessary information for living things to survive and reproduce. Determining the sequence is therefore useful in fundamental research into why and how organisms live, as well as in applied subjects. Because the key importance DNA has to living things, knowledge of DNA sequences is useful practically in any area of biological research. For example, in medicine, it can be used to identify, diagnose, and potentially develop treatments for genetic diseases. Similarly, research into pathogens may lead to treatments for contagious diseases. The goal of exome sequencing approach is to identify genetic variants that alter or change protein sequences, and to do this at a much lower cost than whole genome sequencing. Since these variants can be responsible for both Mendelian and common polygenic diseases such as Alzheimer's disease, whole exome sequencing has been applied both in academic research and as a clinical diagnostic. Dear students, it was all about today's lecture regarding sequencing strategies, PCR-based Sanger sequencing to exosome sequencing. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.